Thank you very much. Now I will talk about not the structural, but now the functional MRI, and I will show you some results on the newly emerging methods that is called the time result fMRI analysis. Just give you a quick overview. The conventional fMRI experiments were heavily relied on functional segregation idea, where distant brain regions were associated with specific functional roles. With the introduction of mostly the resting state fMRI, we start developing newer tools, and we have the functional integration idea, where we have networks that are consisted of multiple brain regions. So one way to recover those networks is very conventional way, depends on the temporal correlations, which is actually the second order statistics. So you can do it in the seed level, where you get here the default mode network, or you can do it in all voxels and then segregate your functional network. So you will have distant networks that have different functional connectivity profile. Or you can do it in many various ways using bilinear decomposition methods, where you represent the whole fMRI data in terms of networks and their associated time series. So now we call all these methods refer them as static because they are driven from the whole fMRI run. And in the literature, it's heavily studied, like how disturb their special patterns in the presence of the disease. So now the emerging perspective of this is the time result models. So here you see two voxel time series of two brain regions. So we can do the same thing as we did the static functional connectivity, and then we can just compute temporal correlation on a time varying manner. And we have seen many talks in this conference about this. So we can drive our dynamic functional connectivity using the second order statistic or other statistic corollary to that. And we drive our connectivity states or time varying dynamic models if we want to do graph analysis. Another way to do represent these time result models is actually on the instantaneous activity itself using the first order statistics. And a very nice example that we also heard that talk about this is actually the CAPS method, where you simply threshold your ongoing activity signals, and then you pick those time frames and you temporal cluster of these ongoing activity time series, and then you get spatially overlapping but temporally segregated activity maps. So what we try to contribute in this field is just adding a hemodynamic information into this model. Actually, everybody knows this hemodynamic response function is in the generative model of the fMRI signal. So instead of doing the temporal clustering on the ongoing activations, we propose a method to do a special temporal deconvolution of the fMRI data, and where we get some information about the transient activity that represents the onsets and offsets of the intrinsic events. And when we do temporal clustering on those transients, we call them innovation-driven co-activation patterns, ICAPs. We got spatially and temporally overlapping networks. So how do we find this? I will just give you a schematic about how we recover those transients through total activation. So just for a while, think of a GLM analysis, where you see measure your noisy fMRI data, and you have a design matrix where you put the regressors of interest. So basically, you know your fMRI data and your design matrix. What you are trying to get is the activation map that is related to the design matrix that you put. So in terms of total activation, we are trying to find actually the regressors without knowing the regressors. So that is what we are trying to do. And we do this in terms of a denoising scheme, so where we only know the fMRI data, and we are trying to estimate these denoised activity-related signals. But inside of these activity-related signals, we use the hemodynamic information in such a way that when you apply the inverse of the hemodynamic response function to the activity-related signals, you get like paradigm-like information that you would put in your GLM model. The nice thing about these activity-inducing signals is actually if you just compute their derivatives, you get sparse innovation signals that represent the onsets and offsets of these intrinsic events. So just to give you a representation, so this we do in every voxel time series. So this is a representative from real data, both signal from a voxel in PCC. And that is what it looks like when we run total activation. So the activity-related signal is the denoised version of the activity-related signal by using the hemodynamic model. So when we apply the inverse of the hemodynamic model to that, so this is what the activity-inducing signal looks like. So it goes ups and downs. And then the innovation signal looks like this. So here, you see the transients where the peaks of these innovation signals, they represent the onsets of the events instead of the ongoing activity. So we went further, and then what we did is we extracted the whole activity-inducing signal of all voxels in the brain. And so these are representative voxels, 3 voxels. And what we did is we temporally clustered the transient. So every transient is assigned to a cluster, and each cluster is actually a special map. So then we use those cluster maps, and we back-projected them on the activity-inducing signal, because that is the real fMRI signal. And then what we have is, for every network, we have both spatially and temporally overlapping activity. So for all these time result models that I just discussed, either the functional connectivity or dynamic functional connectivity or ICAPS or CAPS, so we still have vast amount of time resolved information. So we have to find a way to summarize this time resolved information. So I will just briefly discuss the idea of this. So one of them, we can do some observational measures. So for each state that we have, we can just count their durations, their occurrences, how frequently they switch between each other. Or we can just build another computational temporal models on top of each other. So we have seen a couple of examples of this already. So we can have autoregressive models or hidden Markov models. So for example, if you do a hidden Markov model on the transient or on the activity time series themselves, or we can do some information theoretic approaches as well. So here we show an entropy minimization between the brain regions. So that is good. We have all these advanced tools, and exactly my point is, what type of information do we get from time result models? So I will show you an example from the healthy state first. So this is what you would do if you have a PCC-based, seed-based correlation functional connectivity map. So you will get the canonical default mode network activity. So if you, for example, use ICAPS, instead of getting one network, actually you will get 7 different networks that are somehow representing the default mode network. So that is one thing. So we are actually even increasing our data size and information that we get. So what we see is these ICAPs actually highly spatially overlapping, especially on the posterior parts of the region. So these are the hub regions. They're also highly temporally overlapping. So that means that on average, for this data, for example, we have on average of 3.5 networks active at each time point. So we can further go even more detailed analysis, and we can study the interactions between 2 networks. So this is, I just want to give an example. This is the temporal coupling between the full DMN default mode network and the attention network. And what we see here is actually in all ICAP combinations where these 2 networks appear, they always appear with the same sign. So they are actually positively correlated. So if we look at the another ICAP, which is representing the posterior default mode network, and then we see for all subjects, it's consistently negatively activating with the attention network. So what this tells us is actually a little bit corollary to what is coming on the emerging conceptual dynamic frameworks for mind wandering, that states not all subnetworks within the default network seem to be involved in spontaneous thought to the same manner. So I guess what we should eventually do is, combine find a link between these conceptual frameworks and then using these new conceptual frameworks, build better models. But keep in mind that they should be driven from those. And then using our advanced methods, when we drive something, we should revisit the conceptual framework. So that is a feedback and feed-forward relationship between the concept and the method. So I just want to give you an example of what happens to the dynamic measures in terms of the disease. So here is an example of mood disorders, that 8-minute resting state. So they are mostly major depression and hypomanic subjects. So we recover 19 stable ICAPs. Here they are ordered with respect to their average duration. And what I just want to show you again is we find 5 different components that are part of the default mode network, and all of them has different temporal characteristics. So in the literature, most of the time, in these mood disorder subjects, the hypothesis is actually disrupted spatial patterns of default mode network and emotional regulation networks. So when we compare the spatial patterns of ICAPs between the patient and controls, actually we didn't find any group differences. So that suggests actually maybe there's disrupted temporal dynamics. So this is the result that we get. So the occurrence of some specific ICAPs, which is the insula, amygdala, and posterior DMN, is excessively present, excessively occurring in the mood disorders. So instead of spatial patterns, we here now start seeing the disruptions in the temporal dynamics. And we further checked on the temporal couplings of these ICAPs, the percentage of the sign-dependent temporal overlap. So here we see posterior default mode and the full default mode coupling is severely reduced in the patients, and the same is true for the amygdala and insula. So that is really interesting because all these networks excessively occur in the patients, but their coupling is severely reduced in patients. So final slide in the relevance to the clinical measure of these durations. So if you look at the full default network, actually it relates to the pathophysiology of the disease, and patients' average duration of this full default network correlates with the depression score. If you look at the insula and the posterior default node network, they mostly correlate with the trait-like behaviors, so in the continuous spectrum. So that basically says there's dissociative roles of this default mode subnetwork, posterior versus the full default mode network in mood disorders. So just to summarize, so there is this emerging dynamic field that is coming up that provide very rich spatial temporal information. It can be either on the correlational or the activity level. We should somehow summarize these measures. We can use either observational or further computational temporal models. However, these models should be linked with the conceptual frameworks, and perhaps we need further investigation to close the gap between the computational models and our methodology. So thank you.